Hello, everyone. Uh, Let me get loaded. All right. View. Am I loaded, John?
Uh, let's see. So my slide is up, which is bad. No, wait, I see you're viewing Andy's screen. Yes.
All right. Do
It
you
looks
see
a
my
little
slide?
dark though.
Is it dark?
There you go. Well, that was no. Keep working.
I think I know what the problem is. Is this one? All right. How about now?
That's much better. Yes.
All right. All right. Great.
Promising.
All right. Good morning or good evening, everyone. Welcome to my presentation on restoring a complete pyruvate dehydrogenase complex to syn3B. And thereby increasing the growth rate as predicted by a whole cell FBA computational model. This is work done in collaboration with folks at the Lothé-Scholten group. So since I'm the first talk, I will give a really brief introduction to syn3 and its recent derivatives. So, let's see. Why is my... Oh, here we go. So syn3 is a near minimal organism. Syn1 is a synthetic version. Sorry. Oh. So Syn1 is a synthetic version of natural Mycoplasma mycoides.
Mute yourself until you have a question.
All right. And Syn3 is a minimal version of this. It has around half the size of the genome. It only contains the genes that are absolutely essential for life and some quasi-essential ones to make sure it can, divide in a reasonable manner. As you might expect, it's a not very healthy organism. It has a much slower doubling time, and it makes morphologically irregular cells. Here you can see pictures of the Syn1, which makes nice little regular cells, healthy-looking cells, and Syn3, which makes these huge vesicles that don't look very healthy at all. Then I'd like to introduce Syn3A, which is Syn3 plus 19 genes. John talked about this very briefly earlier. James Pelletier will talk about this, I believe, next week. Here, with the addition of 19 genes, the cells restore their morphology. We get, again, nice little cells that look much more stable. And we have a great improvement in growth time. It goes from around 180 minutes to around 105 minutes. Then the next cell, the next cell I'd like to introduce is Syn3B, which is an improvement from Syn3A, in where we make the cell much more easier to engineer. So to engineer Syn3A, one would have to go through yeast, where one does a cut with CRISPR of the Syn3A genome and uses homology-directed repair to include a gene, and then transplant this from yeast into M. capricolum, which would then turn into a Syn3A organism. Now, this whole process might take around a month. With Syn3B, one can create a donor plasmid in E. coli, which contains a given backbone and an inserted gene of interest, which one can insert with your favorite method of cloning. one can then transform this plasmid in syn3B, and then using Cre-Lox recombination, this gene of interest will automatically get inserted into the syn3B genome, which is a much easier approach. One just has to do a two-piece cloning in E. coli, a plain transformation, which is very simple, and that's it. I'd like to go in a bit further into syn3B. Here we go. As to how this addition of genes works with the LOX recombination. So in the syn3B genome, we have a so-called landing pad, where we have two LOX-P sites flanking a region that can be replaced. And on our donor plasmid, we have two LOX-P sites in which we have our gene of interest and a selection marker. And this whole piece can get swapped into here using a Cre recombinase, which is on the plasmid. And these are mutated LOX-P sites in such a way that it's a one-directional transfer. So it gets transferred once, and then it stays there. And then you can select for cells in which this transfer happened using the selection marker. We usually use pyromycin, but you can also use gentamicin. So that's a quick intro into syn3B and how you can use it to very easily add and quickly add genes to the genome without having to go through yeast and doing transplantation. Now I'd like to go on to some of the recent work that we've been doing. So, in 2019, a whole cell FBA model has been published for syn3A at the time. Syn3B didn't exist yet, as far as I know. And this has been refined over time. And we noticed a little part in the central carbon metabolism where there is some room for improvement in this metabolism. So here in gray, you can see glycolysis going down to pyruvate, and then pyruvate can take two different paths in syn3B. Either it can secrete the pyruvate, or it can convert it to lactate, thereby making NAD and secrete the lactate. There's this arrow here, which is not present in syn3B. It's present in syn1, in which pyruvate can get turned into acetylphosphate while making NADH here. So while if it goes through this pathway, then here NADH is made. Anyway, acetylphosphate then can acetate kinase can then transfer the phosphate on acetylphosphate to ADP, thereby making ATP. Now, ATP is a very is in very high demand in syn3B. So this extra production of ATP here should give the cells a considerable boost in growth rate. So if we simulate the reintroduction of this arrow here in a plain FBA model where we maximize growth, then the FBA model suggests that the doubling time should decrease from 96 minutes to 61 minutes, thanks to this additional ATP getting produced. Now, we constructed this strain using the simple transformation method with syn3B, where we reintroduced this PDHE1 reaction, and we indeed measured a significant increase in growth rate from syn3B had 103-minute doubling time, and syn3B with this one extra arrow had an 86-minute doubling time. So this was very significantly a faster growth. It isn't as much faster as the FBA model predicted, but it's close. So this we view both as a confirmation of our FBA model, and we now have a cell that grows even better than syn3B. So this is another step in making this organism more tractable. I think my time is up.
I think you're about done, Andras.
So thanks for listening, and let's go on with the next talk.
Well, no. We have
No?
a couple of minutes for questions.
Oh,
So
all right.
anyone, just speak up.
Andras, beautiful talk.
Thanks.
Curious
Identify yourself, James.
Oh, hi everybody. My name's James Pelletier. Really excited for this meeting. I noticed in the map of the metabolism that NOx was also shown in red, and I'm curious whether syn3B retains the NADH oxidase or if that's also absent. And I'm also curious
This is
about.
present. This is present.
Okay.
And as you can see, it uses oxygen in order to remove the H from the NADH. And the more flux goes through this green pathway as opposed to going through the lactate pathway, the more oxygen the cells actually need. And I don't really know how much oxygen they're actually getting, but I have a feeling that this reaction may be limiting the amount of flux that can go through this pathway here, as opposed to going through this pathway.
Cool. Thank you very much.
Mm-hmm.